Hi, my name is Jacob McEwen, and I graduated with my bachelor's in the science of nursing in 2017. Since then, I've been working as a nurse on an inpatient psychiatric unit and also on a critical care unit. I am returning to college now to get my last couple of pre-medical prerequisites out of the way in order to apply to medical school with the ultimate hope of becoming a neurosurgeon. Today, I will be presenting the paper Molecular Mechanism for Antibody-Dependent Enhancement of Coronavirus Entry by Juan et al. This paper was initially published online in February of 2020 and ultimately was published in the Journal of Virology in March 2020. This paper is about the phenomenon known as antibody-dependent enhancement of viral entry, which is a mechanism by which antibodies actually facilitate the entry of viruses into host cells rather than preventing it. As an example of why this mechanism can be clinically relevant to humans, the authors discuss the dengue virus, which is a type of floppy virus. Humans who are initially infected with the dengue virus become ill and ultimately recover when their adaptive immune response produces an antibody that neutralizes the virus. But subsequently, if they are infected with a different strain of the virus, the antibody is able to bind to this virus but not to neutralize it, and it ultimately leads to better entry into the host cells by the virus, and the patients become much worse ill than would be initially expected from the initial infection of the virus. The authors discuss that it has been previously known that this sort of mechanism can take place with coronaviruses, and they use the example of the feline coronavirus vaccine actually causing cats to be worse infected by the coronavirus when they are subsequently infected. The aims of this paper are to better characterize this antibody-dependent enhancement of coronavirus at the molecular level with the hopes of benefiting future antiviral and vaccine trials. The coronaviruses are a family of large positive stranded RNA viruses that are known to infect both animals and humans, with two notable and lethal strains of the previously known of coronavirus being the SARS coronavirus 1, which caused an outbreak in China in 2002, and the MERS coronavirus, which caused an outbreak in Saudi Arabia in 2012, and the SARS coronavirus 2, which is currently causing the pandemic, as well as two strains of the coronavirus which are known to cause the common cold. Coronaviruses are so named because when they are looked at under electron microscopy, they sort of resemble a crown due to these protein spikes that radiate out from them, corona being the Latin word for crown. These protein spikes are actually used to bind to host cell receptors and to make access into host cells. These spikes are composed of three S1 subunits bound to a trimeric S2 membrane fusion stalk. Whenever these spikes are able to make access with the receptor protein that they are targeting on the host cell, they're caused to make a conformational change by this binding. They can be found in two different states, either the lying down state or the standing up state. And by associating with the host cell receptor, they're stabilized in the standing up state, which enables the host proteases to cleave the protein in two different spots, one at the S1-S2 junction and one within the S2 domain. This ultimately causes the S1 and S2 portions of this protein to disassociate from one another, and it causes the virus to be able to fuse with the host cell. This ultimately causes infection. Different coronaviruses target different host cells receptors, with the SARS-CoV-1 virus targeting the angiotensin converting enzyme 2 receptor, which is the same as the SARS-CoV-2 virus binds to, and the MERS coronavirus being shown to bind to the dipeptidyl peptidase 4 receptor, which we'll subsequently call the DDP4 receptor. The authors have previous experience working with the coronavirus, and they had actually developed an antibody called Mersabal, which binds to the MERS coronavirus. It has been shown, they were able to show using enzyme-linked immunosorbent assay that the Mersabal antibody was able to bind to the virus in the receptor binding domain. They then subsequently used cell flow cytometry in order to show that cells that express the DDP4 receptor were able to be bound to by MERS-CoV-2 with or without the presence of Mersabal, and that cells that had another type of receptor called CD32A, a receptor type associated with the immune system, were unable to be binded by the MERS coronavirus without the presence of mersabal, but subsequently, when they were exposed to mersabal, this was able to make access into the cells. And by this, they showed that there could be a novel mechanism for the virus being able to get into cells in the presence of mersabal, thus showing that it does cause anti-dependent enhancement of viral entry. Another way that they demonstrated this was they took a certain type of kidney cell and they caused them to express 3 different types of receptors: CD16A, CD32A, and CD64A. These are different types of immune system associated receptors, and it was shown that in the presence of mersabal, viruses were again able to make it into the CD32A receptor, but in the absence of mersabal, they were not, and they also, either way, didn't show activity for the other subtypes of the immune system receptor. In order to further enhance these lines of evidence, they also exposed receptors or cells that had the CD32A receptor to SARS coronavirus 1, both in the presence of and in the absence of a certain type of monoclonal antibody 33G4. And in the presence of this antibody, it was shown that SARS-CoV-1 can make access to the CD32A receptor, but in its absence, it could not. Although in both cases, the mersabal and in this other monoclonal antibody, the respective viruses were inhibited from making it into their viral receptor. And this shows that these antibodies, although efficient in blocking the virus from getting into the receptor that they originally intended to, through the same binding domain, can actually enable them to make it into different types of receptors that they would normally not be able to. Another way that they demonstrated this mechanism where they took the MERS coronavirus and they exposed it to cells that express DDP4, and they showed that in the absence of DDP4, that the coronavirus was able to be cleaved by trypsin at one site, and thus was not activated. But in the presence of DDP4, the trypsin was able to cleave the protein in 2 different sites and thus activate it, showing that it was through a conformational change that the S2 site was able to be exposed for cleavage, and further demonstrating the mechanism by which this virus makes it into cells. It was shown that the same mechanism is true of mersabal. If you take the virus and you expose it to mersabal, it was able to be exposed at the S2 site, and thus there were 2 cleavages as well. The authors also looked at the virus via negative stain electron microscopy as it was bound to the mersabal, and they showed that it is, and through this they were able to show that it is actually in the receptor binding domain that mersabal binds the virus. Unfortunately, due to limitations with the resolution of this sort of microscopy, they're unable to tell the exact conformation of the protein, but because it's been shown previously that the receptor binding domain can only be bound to when the virus is in the standing up position, they presumed that that's the conformation of the protein. Ultimately, the proteolysis studies and the microscopy studies confirmed were in unison in confirming the results. They also took the cells that were caused to express DDP4, CD32A, or both of these types of receptors, and exposed them to different degrees or levels of mersabal. And they were able to show that as the level of mersabal increased with the cells that only express DDP4, the ability of the virus to actually make it into the cell decreased. When they exposed cells that express CD32A to mersabal, initially as mersabal concentrations increased, the binding of the virus increased, but then subsequently it declined. And the authors hypothesized that this is because as the mersabal saturates the receptor sites, eventually it gets to a point where the indirect contact between the virus and that receptor site is inhibited. And the same thing was shown if cells both express the CD32A and the DDP4, showing that the same mechanism is probable in those cells as well. And eventually there becomes a point where the presence of their actually goes on ahead and inhibits even at the CD32A site. What this means is that an actual in vivo different concentrations of viral receptors and immune system associated receptors on the outer membrane of cells would cause a complex interplay, and depending on which one predominated would depend whether or not the virus was able to use this mechanism to make it into cells. They gave the example that transgenic mice caused to express the DDP4 receptor that is seen in humans were actually successfully protected from the coronavirus after exposure to the vaccine because the mechanism by which it actually blocked the DDP4 site was more potent than the mechanism that enhanced the CD32A site binding. And so the authors successfully did increase our knowledge about the actual molecular mechanism for the binding of these viruses and their enhancement because of the monoclonal antibodies. Another advancement that was made by this paper is that the authors were able to show that the antibodies they used here were actually produced by cells that had initially been exposed to the same strain of the virus, whereas previously this mechanism has been shown when a different viral strain is introduced to a host that has already been exposed to a unique viral strain from the one that they're being exposed to when the antibodies caused the enhancement. So ultimately, this research has relevance to vaccine development as well as antiviral drug development, because if it was shown that the mechanism by which the enhancement takes place is through the virus being bound at the receptor binding domain, and subsequently we should try to make vaccines or antiviral drugs that do not bind to this domain. And if we do so, we have a greater chance of inhibiting their ability to make the virus ability to make it into host cells without enhancing the binding. This could have implications for reinfection. So if it's shown that SARS coronavirus 2 can cause reinfection, there could be worsening infection by a similar mechanism. And by better characterizing how this mechanism takes place, it enables us to be on the lookout for that and be able to look into this mechanism further. The authors said that there needed to be further research in vivo because of the complex interplay of the different receptor types, and further characterization of what dosages would cause antibody-dependent enhancement actually in a living model versus just the in vitro studies. So thank you for taking the time to listen and watch this.